physiology. We're getting down to it here. We're into the last couple of chapters that we're going to be doing this semester. So what we're going to be working on for the next week or so here, next couple of weeks, is going to be the nervous system. Similar to the muscles, a little bit more involved on this one. Definitely some of the physiology is going to resemble what we talked about before. So these ideas of action potential, membrane potential, a lot of that is going to be, in all honesty, similar to what we talked about with the muscles. So to start with, though, we're going to talk about why we have a nervous system and kind of what are some of the main parts of that nervous system. We're then going to get into how do we generate nerve impulses, how do they travel within the cell, how do they travel between cells, and then finally towards the end here we're going to talk about how do the different cells go about actually interpreting these different nerve impulses. So how does any particular cell know what an incoming message it means? And how can we send information with these incoming messages? So the first one here is, why do we have a nervous system? In a simple sense, it is one of these command and control type of systems of the body. So we have billions of neurons that are all linked together, and it is a command and control system of the body. So integrating a lot of these different things that are necessary for homeostasis, so detecting stimuli, generating responses, things like that, really involved in regulating most other body functions. This, along with the endocrine system, are really what are doing most of the command and control of the body. The other thing is the nervous system is really your personality, is what makes you you and not just what makes you distinct, I guess, the simplest way to say it. So in terms of the basic functions of the nervous system, this is one of the actual simpler sets of functions compared to a lot of the other body systems. Nervous system is really doing three main things. Sensation, so we have stimuli that we're aware of as well as stimuli we are not aware of. The body is monitoring a lot of this using different receptors. The other thing that the body does is it does a lot of motor output. So we send signals to the muscles, to glands to trigger muscle contraction, to trigger release of a chemical. Those are those motor outputs. That's a reaction. Really what's happening in between those two is the other main thing. This is that integration or processing, association, if you want to call it that. This is a lot of these, the central nervous system, as we'll see what that is in just a little bit here. This is doing a lot of this processing to determine what the sensory information means and how the body should respond to that. So it is very similar, like I said, to the endocrine system. They are both command and control systems. They both monitor stimuli, react to the stimuli. They both actually use chemicals. The main thing is the nervous system. It's a fast-acting one here that uses neurotransmitters across small distances, where the endocrine system is going to use blood-borne chemicals called hormones that are generally going to take longer to bring about its action, and usually you're going to have much longer-lasting effects. And then what we can look into is a lot of these different subdivisions of the nervous system. We really talk about two main initial ones here. You have the brain and spinal cord, which we refer to as the central nervous system. This is where all that integration takes place, where the control part of the body is for the most part. We then have the peripheral nervous system, which is the combination of the 12 cranial nerves, or pairs of cranial nerves, and the 31 pairs of spinal nerves. These are taking that information from the central nervous system to all areas of the body. And then a lot of these, we can subdivide this down even further, and you probably notice this with A&P. We do a lot of dividing and then subdividing and then subdividing. The peripheral nervous system, you really can talk about it in the sensory versus the motor. Sometimes referred to the sensory as the afferent division, the motor as the efferent division. The sensory division is taking stimuli and taking impulses from these receptors back to the central nervous system where they can process it. If we are consciously aware of this, it is the somatic sensory. If it is from organs and stuff that we are not consciously aware of, this is going to be the visceral sensory division. The motor division is very similar to this. It is that efferent division, taking stuff from the central nervous system out to these different muscles or glands. And much like with the sensory division, we have these, as you can see here, the somatic sensory. These are ones that you consciously perceive, things like the eyes, taste, touch, body position. We also have visceral stuff here in terms of sensory stuff that we don't consciously perceive, like blood pressure, number of other ones here, blood chemicals, for example. The motor division, like I was starting to say, is very similar to this. We can have as well in here, somatic nervous system. These are the motor division of this, though, is the stuff that we have voluntary control over. So voluntary control of skeletal muscles. And it says generally because there's things like shivering, which is not voluntary. But the somatic nervous system is those voluntary muscles that we can control, the skeletal muscle. The autonomic nervous system is going to be muscles and glands and other things like that that we do not have conscious control over. Things that would maybe influence heart rate, moving stuff through your digestive tract. That is part of this autonomic nervous system. And again, we can subdivide one more time into the subdivisions of this autonomic motor nervous system, which is going to be the sympathetic versus the parasympathetic. So the nicknames on these ones, a lot of times you've heard, usually the fight or flight for the sympathetic nervous system. Parasympathetic is going to be nicknamed the rest and digest, not as well known for most people off the bat. The main thing is these two tend to work against one another. So something that is upregulated by the sympathetic nervous system is generally going to be downregulated by the parasympathetic and vice versa. One thing to realize is sympathetic is not always activating, parasympathetic is not always toning down. Parasympathetic, for example, with the digestive tract increases activity, sympathetic decreases it. When we get into AMP2, this will be one of the first things we talk about in a little bit more detail, is this autonomic nervous system and some of the stuff that's going on with it. So the other part on this first video here I want to talk about is some of the different cells of the nervous system. So when we talk about nervous tissue, we didn't go into much detail on this with the tissues section. In reality, there's two main types of cells. This is generally just cells is the main type of tissue. There's not a lot of space, other things like that. It's very much these two types of cells. So you have neurons, which are the functional nerve impulse conducting cells. These are cells that we can stimulate. They can transmit signals down one of their processes. Those are the neurons. The neural glia cells, there's actually 6 different types of these. These are the non-excitable cells. These are ones that do not send impulses. But these are going to be supporting to those neurons in a number of different ways, as we'll see as we go through them. Again, characteristics of neurons, they are excitable, much like the muscle cells. They can respond to a stimulus and bring about some type of change in there. They're conductive. Again, very similar to the muscle cells. They have electrical charges across their membrane. We can bring about changes to that membrane, and these can be either local or they can be self-propagating action potentials like we saw with the muscle. These cells are capable of secreting neurotransmitters. This is a lot of times how they're going to send the signal from one cell to the next one. The other thing that's very interesting in these ones, these are extremely long-lived cells. A lot of these ones are formed prior to birth and stay alive all the way through to about the time we're dying in a lot of cases. The other thing is most of these cells stop dividing after a period of time. Not all of them, but a vast majority of them. So these cells pretty much become amitotic, meaning they don't divide. So damage to neurons is usually going to be something that's permanent. We cannot replace damaged neurons very easily. And this is why a stroke usually leads to loss of function and other things like that. Like I said, the glial cells, these are found in both the central nervous system and peripheral nervous system. Different cells in each of those ones that there's no cells that are glial cells that we see in both areas. But there is 4 different types we're going to talk about in the central nervous system and a couple that we're going to see in the peripheral nervous system. These are generally smaller than neurons. They divide a lot more often. They make up about half the volume of the nervous system, but because they're smaller, they far outnumber the neurons, about 10 to 1. In a lot of ways, these are going to protect or nourish these neurons, provide structure for these neurons to be attached to, help guide these neurons to where they're going to connect. And in a lot of cases, these are critical for the neurons to work correctly. So these are some of these different types of glial cells. So you can see in the peripheral nervous system, we're going to talk about these satellite and schwann cells, or neuromyelicites is another name for a schwann cell. In the central nervous system, we're going to see that there's 4 main types here. You have astrocytes, oligodendrocytes, microglial cells, and ependymal cells. This is a nice little summary. We're going to walk through each of these one at a time, so I'm not going to walk through this whole slide at the moment here, but this is a good one to come back to if you want to get the main functions of each of these. So astrocytes get their name because they're basically star-shaped, and the ends end in these little things called perivascular feet that we will see in a lot of cases surround blood vessels in the central nervous system and is part of forming that blood-brain barrier. What the blood-brain barrier is, blood does go to the brain. There's blood vessels within the brain, but there is distinct specific layers that surround these blood vessels that are much more selective than we see in other areas of the body, and a lot of things that are in the bloodstream cannot get into the nervous tissue as easily. Astrocytes are part of this. So most abundant by far of these neuroglial cells, like I was already saying, they wrap capillaries or those perivascular feet around that and form this blood-brain barrier, with the exception of a few areas that we'll see in the brain. We'll talk about that in AMP2, but the brain and the nervous tissue is very selective on what's allowed into these tissues and what is not. This means a lot of chemicals and other stuff cannot get past this blood-brain barrier, and it's a protective layer for the brain. The other thing that these astrocytes are doing are helping to regulate what's in this space surrounding these neurons. So they really are regulating the nutrients and other things that are surrounding these neurons, making sure the neurons get what they need for normal functioning. And again, provide a structural network for these neurons to travel through. They also are going to help when the neurons are developing, and if any neurons die, these a lot of times will fill in that space with that, so there's not just a space there, they'll be filled in with these astrocytes. Ependymal cells, these line the cavities within the brain and spinal cord. There's one specialized blood capillary that there's a gap in that blood-brain barrier called the choroid plexus. These ependymal cells line some of that. They help make the cerebrospinal fluid, which is a blood filtrate. And they also are going to have cilia that help circulate the cerebrospinal fluid around the brain as well as up within the spinal cord. Microglial cells are some specialized phagocytic cells. Again, because blood cells cannot leave the blood and get into the nervous tissue, it has its own phagocytes. These are the microglial cells. Not a large amount of them here, but these wander within that nervous tissue and help engulf anything infectious and get rid of it, and can also remove dead and damaged tissues within the central nervous system. And the last one of these ones, it's the one that's the hardest one to say and spell, which is oligodendrocyte. These are cells that have these little extensions that go out and will wrap around extensions of a neuron called an axon. These wrap around this axon, insulate this with something called a myelin sheath, all of which we'll be talking about in more detail. These are the cells that make the myelin sheath within the central nervous system. They allow these action potentials to travel faster and move through the central nervous system easier, and we'll get into why that is the case when we start talking about how impulses travel within cells. So this again shows you all these ones here, shows the myelin sheath, the perivascular feet, you can see the astrocytes, as well as some of the other ones here, the microglial cells, ependymal cells lining the opening right here, the microglial cells here and here. Within the peripheral nervous system, we have two main types here. We have what are called satellite cells. These are around cell bodies in a ganglion, which is a group of cell bodies outside of the central nervous system. These are separating the cell bodies in the ganglion from the surrounding fluid, kind of acting like some of the astrocytes do in the central nervous system. Again, regulating within these ganglia nutrients and waste products. The other cell type we're going to see are the neurolamicites. These are also known as schwann cells. These are doing a lot what the oligodendrocytes do in the central nervous system. These are going to wrap these peripheral nerves with a myelin sheath, and again, much like the oligodendrocytes do in the central nervous system, allow for this insulation of the axon as well as faster propagation of action potentials. So you can kind of see these satellite cells surrounding cell bodies in the ganglia, and you can see how these neurolamicites or schwann cells cover each of these here. Unlike with the oligodendrocytes, so each one of these is a single schwann cell, where you saw with the oligodendrocytes there was multiple extensions coding multiple axons. That ends up being important to us when we start talking about damage to axons in the central nervous system. Those oligodendrocytes can't help retarget this axon back to its connection. Schwann cells can in the peripheral nervous system. So if you sever a nerve in the peripheral nervous system, we can actually regenerate that axon and reconnect that nerve. It might be a slow process, but we're capable of actually doing it. So that was kind of the first one you're giving us some of these subdivisions and the basics of the different cell types. What we're going to pick up with next time is we're going to start talking about the neurons and the extensions and what's kind of going on there. So I will see you next time.